Good afternoon, dear students. Today again we are having a new lecture of biomolecule. In that biomolecule series, we have already covered the lecture number 8. In that lecture, we have already discussed about the formation of starch, glycogen, and cellulose. This time, that carbohydrate is fully completed. This is the lecture number 9 for the biomolecule, and in this lecture we are going to discuss about the protein. Okay, so protein we will cover in two lectures. In first half, 50% protein we will cover, around 20 to 30-minute video, and in the second lecture full protein will be discussed. So at the same time, in the last video, if you will go through it, we have discussed the formation of starch, glycogen, and cellulose. Okay, so this is now we are going to discuss about the protein. So let's talk about the protein. What is protein? So small terms are there. That protein word came from the Greek. That came from the Greek word proteus. And the meaning of proteus is something of prime importance. Okay, so something which is prime importance for us, for our body, that's called protein. So actually, what are the proteins? So proteins are polymer polymers of alpha amino acid. So what I am saying, proteins are the polymers of alpha amino acid. Focus on it. They are polymer of alpha amino acid. Only alpha amino acids are the proteins. We are having other amino acid also, like you can make a note here. Polymers of beta and gamma. Polymers of beta and gamma amino acid also exist. Amino acids also exist, but protein contains only alpha amino acid. Once again I am telling you, this protein topic is very important for the competitive point of view, as for NEET and JEE. So polymer of beta and gamma amino acid also exist, but proteins only contains alpha amino acid. Now talk about the amino acid. What is amino acid? So in the case of amino acid, R, CH and NH2. In that case, this COH is your prime group, prime functional group, or you can say main functional group. Attached carbon with this COH is your alpha carbon. So this is your alpha carbon, and on this alpha carbon we are having amine. And what is the prefix used for the amine? That is amino. So this is the basic unit of amino acid. If we are talking about the natural amino acid, so you can say natural amino acid are L-amino acid. Natural amino acids are L-amino acid. L-amino acid means it's a naming, DL naming. So L-amino acids are natural amino acid. They are natural amino acids. So how to write DL naming, that we have already discussed in the carbohydrate chapter. In that case, I explained how to write D and L naming. Here also we will discuss, but here you have to focus on it. Natural amino acids are L-amino acid. Okay, children? Let's talk about the structure of amino acid. So what kind of structure they are having? Carbon, one hydrogen, and this is your COH group. Dash position we are having R, and the wedge position we are having NH2. So COH and hydrogen on the plane, and that R backside of the plane, and this NH2 towards you. So this is the structure of amino acid. And what is R here? R is called residue of amino acid. Residue of amino acid. Okay, so this is the structure. Let's talk about the different kind of amino acid. One table in the NCERT given, so that same only I am writing here. In that case, for the competitive point of view, 3 to 4 amino acids are very important where we have to focus. So what are these? At the place of R, what you can use, then what is the name of the amino acid, what symbol you can use, and what is the code of amino acid. Okay, so at the place of R, if you are using H, then that amino acid is called glycine, and the symbol is used capital GLY. Code is capital G. At the place of R, if you are using CH3, then it's called L-9, and the symbol is ALA, and the code is capital A. At the place of R, CH3 whole twice CH, then it's valine, VALINE. Symbol is VAL, and code is capital V. Next, CH3 whole twice CH, dot CH2, then it's called leucine, LEUCINE. Symbol is LEU and the code is capital L. At the same time, CH3, CH2, CH, CH3. This you are using at the place of R, it's called isoleucine. And ILE is the symbol and the code is capital I. Okay, so these are the amino acid. Few more again are name, symbol and code. At the place of R, if you are using H2N, CH2 whole 4, then it's called leucine, and LYS and code is capital K. If you are using HCHOO, then CH2 whole twice, it's called glutamic acid, and the symbol is GLU, code is capital E. HSCH2, it's called CYS TEINE, CYS and code is C. Phenyl CH2, phenylalanine, phenylalanine, PHE and the symbol is F. So these are the all amino acid which given in the NCERT, where you have to focus, that only you have to remember. In that case, this glycine is important, this leucine is important, and this glutamic acid and alanine. These are the 4 important amino acid. In these 4 you have to focus and learn. One very important thing here, that glycine is the only amino acid which is optically inactive. Remaining all amino acids are optically active. Glycine is the only amino acid which is optically inactive, because at the place of H, at the place of R you are using H and we are having 2H on the same carbon, so it will be optically inactive. Remainings are optically active. One more thing, these name derived from common name. These names are derived from common name. Okay, so let's talk about the DL naming. How? So once again you can check it. These are the name only. This table is also given in the NCERT. So you have to focus on 4 only: glycine, alanine, where you have to focus glycine, alanine, lysine and glutamic acid. Okay. Let's talk about the naming of amino acid. So what are the rules in the case of carbohydrate, if you remember? So in the case of carbohydrate, once again I am telling you, where you are using capital D, when OH on right side, when OH on right side. Similarly, in the case of alpha amino acid, when NH2 on right side. Where you are using L, when OH on left side. So here also NH2 on left side. You have to check the position of NH2. So example, COH and here NH2, R and H. So NH2 is which side? Right side. Then it is D amino acid. In the second case, NH2 on left side, then it is L amino acid. From bottom to second carbon you have to focus and check the position of OH. That will be your DL naming in the case of amines. In the case of alpha amino acid. So naming is completed. Now classification. Classification of amino acid. So on the basis of character, they are classified into two categories: essential protein and non-essential. What is the difference between essential and non-essential? Those which cannot synthesized in our body those which cannot be synthesized in our body, these are the essential protein. These proteins we have to take from the outside sources like dry foods, pulses and all. Non-essential means those that can be synthesized by our body. Okay, so here these proteins we have to take externally, and this kind of product already formed in the body internally. This is the first classification on the basis of character. Second classification on the basis of acidic and basic nature. On the basis of acidic, basic nature. In that case, they are classified into three categories: acidic, neutral and basic. Okay, so focus on it. What is the amino acid? RCH, C double bond OH and NH2. What is the COH nature? Acid. What is the NH2 nature? Base. So here that acid and base nature depends upon the COH group and NH2 group. Okay, that means if COH is more than NH2, then it is your acidic protein. If COH equal to NH2, then it is your neutral. When COH is less than NH2, means NH2 is more than CH2, you can say, then it is basic. Okay, so that acidic and basic nature depends upon the COH and NH2 group, and like that you can classify. Examples, if we are talking, so acidic amino acid, glutamic acid, and aspartic acid. These are the example of acidic amino acid, neutral glycine and L-9. These are the neutral. Lysine and arginine. These are the basic proteins. So this is the classification. Let's talk about the properties. So amino acids are water soluble. Why they are water soluble? Due to hydrogen bonding. Due to hydrogen bonding in COH and NH2. Second is colorless, high melting solid and crystalline solid. Crystalline solid due to Zutter ion formation. So these are the property, and this is the first of the lecture for the protein. Remaining Zutter ion and all we will discuss on the next lecture. So thank you.